All right, good morning, everybody. My name is Amy Kierstead, and I am an Associate Dean in the College of Arts and Sciences here at the University of New England. And today is our Symposium Day. Today is our 21st Annual College of Arts and Sciences Research Symposium. And this is, I think it's the most wonderful day of the year. This is the day that we get to showcase the outstanding work that our students have done as part of a class project, as part of a research project or scholarly work with a faculty member or individual creative works. And you know, our symposium looks a little different today, but even COVID can't stop us. So we're excited to showcase some of our research projects in this interactive webinar. We hope you'll have questions for us. And it's my pleasure now to introduce the Dean of the College of Arts and Sciences, Dr. Jonathan Millen.
Thank you, Amy. Good morning, everybody. Amy, as you know, this is my first real spring research symposium. It's something I've been looking forward to for a very long time. You probably recall, might have been the third time I met you was watching you and Dean Hay moving about the symposium. And it was one of my first real introductions to the students of UNE. And I was absolutely impressed and overwhelmed and inspired by the work that they've done across the college, across our 6 different schools, across the disciplines. The evidence of students working so closely with faculty on generating research is just, it speaks really to the heart of what we do. And so I'm so excited to be here with you today. Even virtually, I can't wait to see what our students are doing, get to chat with them and show this to the world, the great things that our students are doing. So take it away.
All right, excellent. So our first speaker is Emily Scott. Emily is completing her Master of Science in Marine Sciences. She's working with Dr. Carrie Byron, and she is going to tell us about her project that's titled Ecosystem Services of Seaweed Aquaculture off the Gulf of Maine. So take it away, Emily.
Great, thank you, Dean Kirsten. Okay, so first let's start off with me explaining what ecosystem services are. So these are basically material and non-material benefits that humans obtain from the environment, usually for free. And in the realm of kelp or seaweed aquaculture, we obviously get provisioning services such as food and raw materials. And then there also has been regulation and cultural services work. But however, the habitat and supporting section of this wheel, these ecosystem services, has not been studied that great in kelp aquaculture. So for that reason, that's what I am focusing my research on. Oh, no. Okay, so again, I'm focusing on these habitat benefits. Is the kelp farm providing habitat to organisms that are living in the surrounding area? And doing this will fill the knowledge gaps that are currently present. So I am using GoPro camera visual surveys. I'm also collecting invertebrates, and I am doing environmental DNA, which that's just, you take a water sample and there's all this DNA from shed skin cells or feces from organisms living in the water. You can put it through a bunch of these processes and you basically are able to tell what's living in the water at that time. So this research is important because we are seeing a decline of wild kelp forests here on the Gulf of Maine, and possibly we could use kelp and seaweed aquaculture as a temporary way to balance out these ecosystem services that we are losing because of the decline of the wild kelp. So, and then also it's just important to evaluate whether or not the kelp is harmful or if it's helpful to the environment. And then my results, I've collected now over 115 hours of GoPro video footage. And the only thing that I have seen is a seal and the sea robin here. I think we're not really seeing much because the water is very cold right now. It's only like 40, 45 degrees. So maybe if we continue this into the summertime, maybe we will start to see more organisms. I have not analyzed the invertebrate collections or the eDNA yet, and that will be this summer. I will do that this summer. So.
Emily, that's fantastic. Can I ask you a couple of just real quick questions for me?
Sure.
Help me understand kelp related to seaweed. Are those two synonymous, or is there something about kelp that's unique and different?
Yeah, so basically, when you talk about seaweed or algae, you have red, brown, and green, and only brown seaweed is considered kelp. So
And
that's
is it the chemical composition? Is it something about the?
It's just the
Just the color?
genus or the species, yeah, that makes it that color, that puts it in that realm. Yeah.
And a second question, if I heard you, you said that you were looking to see if the kelp might be dangerous.
Yeah, I mean, dangerous, I think that's kind of a hard word. It's just, we're trying to see if the kelp is providing habitat for these organisms. And it's not a problem that we're not seeing organisms right now because the water is so cold. Because this kelp grows over the winter time. So we've only had positive research for kelp. So.
And Amy, can I sneak one more quick question in?
Absolutely. I'm not seeing anything in our Q&A, so lots of opportunity.
But you know me, I'm just so curious about these things. So I know that you've been using our farm off of Rams Island, and how does that help the research? How would it be different if you weren't studying it on the farm itself and just studying it in the wild?
Well, in the wild, right, we know it's providing habitat services. That's been known for a long time. So being able to have the kelp aquaculture places right off school, it's so convenient and easy to study for that. Like, I don't know any other school that has these resources. So doing my master's here, that makes it really special for me because I'm just able to go out and use the farm at Ram and then also at Wood too. So
that's great. And I didn't pay you to say that, right? That wasn't the rehearsed.
That's totally, no.
No, that's great. Thank you, Emily.
Yeah. Thanks, Emily. That is so interesting. And we're so pleased that you're doing your master's in marine sciences here. And, Dean Millen, as you know, we have master's programs in marine sciences, in biology, in environmental science. And we also have our professional science masters in ocean food systems. And that's a unique program. And so we've got some excellent work going on with our master's students in the research area, but we also have a very rich and vibrant undergraduate research program. And so now we're going to pivot, right? The word of the 2020s, pivot. We're going to pivot to hear from some of our undergraduate research students. And our next student is Nicole Martin. Nicole is a graduating senior majoring in psychology. And she has been working in Dr. Stigler Balfour's lab, research lab, the Reading Comprehension and Cognition Lab, since her freshman year. And she's going to tell us about a special project that was inspired by the pandemic, which is called The Psychology of Zoom: How Lecture Delivery and COVID-Related Stress Impacts Student Learning. So take it away, Nicole.
Thank you so much, Dean Kirsten. So so sorry, my cat will not get off my lap. So the COVID-19 pandemic, as we all know, has changed life as we know it. Social distancing brought distance learning into the spotlight, and professors had little time to adapt their content in the shift from a digital classroom, from an in-person classroom to a digital one. So our research was really interested in better understanding how these online lectures impact student learning, but also how they're presented impacts student learning. We were also interested in better understanding the role of student characteristics facilitating their ability to adapt to this new delivery method. So for our study, we presented students with an online lecture that was 10 minutes long. We had 234 students watch this pre-recorded lecture. And for all of the videos that they watched, the content and the PowerPoint was exactly the same. It just changed based off of how the video was presented. So in condition 1, students could see the lecture, see the PowerPoint, and hear the professor talking, but they couldn't see the professor at any point. For condition 2, they could see the professor in a small video throughout the entire lecture, And for condition 3, they could only see the professor at the beginning and the end, and they could see her full screen, but they couldn't see her at any time when content was being presented. So after they watched this lecture video, they took a quick recall survey and then answered a whole bunch of other survey questions to measure the personal variables that we were interested in exploring. So that included mind wandering, how focused they were on the task at hand, social presence, how much they felt the professor was actually there with them, talking to them, cognitive load, how able they were to take on new information, fatigue, how tired they were, and COVID concern, how worried and stressed out were they about COVID at that time. And so our results ended up finding that delivery did matter. The students who saw the video with the talking head present throughout the entire lecture performed significantly better on the recall quiz than the students in the other 2 conditions. But almost more interestingly, we found that those personal factors really did matter in determining students' score. So we saw that 3 of the personal factors directly impacted their score, and that was the social presence, mind wandering, and cognitive loads. But fatigue and COVID concern had an indirect but pretty big impact on the score through that cognitive load. So again, fatigue and COVID stress are impacting how much new information students are able to take on, which really shows us how much students were impacted by the pandemic. Not only was their learning environment completely shifted, but they were being greatly impacted by the situation they were presented with. So overall, the study shows us that while yes, it's important to keep the external distractions seen in the lecture presentation low, we want to keep those external distractions low, those internal distractions are just as important. So for my professors who are listening, that small video in the top corner of your pre-recorded lectures, it's really helping students out. But on the other side, don't forget that those personal factors are impacting students above and beyond for their performance in the classroom than anything that lecture format can do.
Thank you, Nicole. How are you?
Good, how are you?
Good to see you. So I have a question. Did you, or were you able to account for whether or not there was a cat in the room when the students were studying and whether or not that might reduce the stress of the learning environment?
Again, I'm gonna say from personal experience, it's very distracting.
I'm just kidding, of course. Did you look at other factors like synchronous versus asynchronous and whether or not the students were watching the video as it was being recorded as opposed to watching it on their own time? And might that factor into your findings at all?
Yeah, it definitely might. All of our whole study focused around asynchronous learning, just because for us, logistics-wise, that was really what our lab could do most reasonably. So all of our stuff was focused on asynchronous. And while there are some confounding variables, such as like time of presentation and like the professor being able to see you or not, that we think might play an impact, we do think that some of the stuff would probably carry over to a synchronous lecture.
Now, Nicole, as you know, we moved to a hybrid model because we had to. And over the past about a year and a half now, faculty have been experimenting with different modalities and technologies. What would you recommend moving forward? You know, presuming in the fall that we're back face-to-face and we no longer have to rely on recorded lectures, is there a place for that, do you think, to sit side by side face-to-face, or do we just use it for emergency situations?
Yeah, I mean, for sure. I think our research probably lends itself well to the many students who do online learning, regardless of if we're in a pandemic or not, because as we know that digital learning is increasing even outside of the pandemic. Maybe not for 20-year-olds, but you know, there are a lot of people who go to school outside of that demographic. In regards to combining with face-to-face, that's definitely not something our research touches upon. But I mean, as a student, I could say I know that I've found some of the videos to be super helpful because you can go back and rewatch it, especially on some of the like upper level courses that are really, really challenging. Having those videos can be super helpful because you can rewatch the same 5 seconds over and over again until it finally clicks in your head. So that's not related to my research, but my personal experience, that's what I would have to say.
And Amy, can I get one more in there?
You can, although we have a question in the live Facebook chat.
Let's defer to that.
All right, well then that's a great opportunity for me to remind folks that if you're watching on our live stream, you can also engage with us on our Facebook Live, University of New England, and ask some questions there as well. And hit the like and the love button as much as you want as well to cheer on our students. But this question is, do you have any recommendations on what students could do to help with their COVID repeated stress and fatigue to increase their ability to take on new information?
Yeah, that's a great question. It's definitely not something that our research is looking into for our lab, because we do focus on more of like the cognitive experiences that students are having. But as a student myself, I would probably remind my fellow students to, take some time for yourself, take some time to de-stress and decompress. Even though we're in the middle of a pandemic, that self-care is still just as important as any other school year. While it might look a little different because we can't just go out for coffee with some of our friends, still make the time for it, whether it's doing a little breathing exercise before you get to class, maybe putting your cat in the other room so they don't distract you during the presentation. Just make that time for yourself so you can be as present as possible and prepared for those classes you have.
That's wonderful. Thanks so much, Nicole.
Yeah, thank you, Nicole. Nicole, you were one of the first students that I met when I arrived at UNE, and I was immediately impressed by your general enthusiasm, but the seriousness with which you approach your scholarship is so impressive. And we learned today from Dean Kierstead that you've really been doing that since your first year. And that's a hallmark of the UNE experience. So Dean Kierstead, how is it that students can get involved so quickly, and how do those opportunities avail themselves to our students?
Yeah, absolutely. So there are different ways for students to get involved with undergraduate research. Usually it means making a connection with a professor, and that can start right with your faculty mentor or a professor that you have in one of your first year classes. Oftentimes students will start by volunteering. Maybe they respond to an ad for a work study position. And those first positions aren't that glamorous. Sometimes you're washing dishes or, doing what we might call grunt work. But it's a way to sort of get your feet wet, if you will. And then from there, students will often, earn course credit for engaging with their research projects. We have a program called the Summer Undergraduate Research Experience, or SURE. And Nicole, I know you and many others on this presentation were part of that, where students can stay here over the summer and really dive deeply into their research projects. And of course, I'm going to give a plug on Nicole's behalf for our student-run Research Experience Club, which is another great way to connect with students who are engaged in research and scholarship across the university. So certainly lots of ways. And if folks have questions, please reach out to me. I'm happy to chat more.
Thanks, Amy.
Yeah. So we're going to move to another student who's been involved with undergraduate research. In fact, before she even formally started at UNE, she was volunteering in Dr. Kristen Burkholder's microbiology lab. And that's Katie Rusa, who is a medical biology, medical sciences graduating senior. And she's going to tell us about her project, which is titled Pyrogalol Impairs Biofilm Formation by Inducing Bacterial Oxidative Stress. Go for it, Katie.
Okay, so hello everyone. I'm Katie, and I work in Dr. Burkholder's lab, which is in the School of Biological Sciences. And my research focuses on understanding and mitigating bacterial infections, which is basically understanding how bacteria cause infections. And some of our pathogens of interest are drug-resistant Staphylococcus and Pseudomonas bacteria. And that's what you see on the right hand in the picture. That's Pseudomonas, and it's one of the most common hospital-acquired pathogens. Okay, so why are we doing this? So as some of you might know, antibiotic resistance is a worldwide problem because existing antibiotics trigger bacterial drug resistance by killing susceptible bacteria and leaving those that are resistant against pathogens, against the drug behind. And so new anti-infective drugs are needed to combat infections by anti-infective drugs are needed to combat these drug-resistant infections. And so a new strategy for developing antibiotics is to use drugs that inhibit infectivity of the bacteria without directly killing the pathogen. And so because the majority of infections that are caused by microbes that form biofilms, those are thick, slimy films that stick to the body and on medical devices. And so that's what you see in the bottom right corner. That's how biofilms are formed. And in the top picture, that is from the NIH of Staphylococcus aureus. And so these biofilms actually exacerbate infection and that make them very difficult to treat. So our goal is to find ways to block biofilm formation as a way to reduce their infectivity. And we have identified compounds that block biofilm formation in Staphylococcus and Pseudomonas, which are, like I said, the two most common hospital-acquired pathogens. And so we studied the effects of several phenolic compounds on biofilm formation, and we found that the phenolic compound Perogalo, which is in the left top corner, reduces biofilm formation in the two species of Staph and Pseudomonas. And so this is exciting because these, like I mentioned, cause the majority of bacterial infections in hospitalized patients. And these compounds block their most important mechanism of infection, which, like I mentioned, is the biofilm. And so you can see the three graphs that we have at the top. They're showing as we increase the concentration of our compound, we are actually reducing biofilm formation. And so my research focuses on understanding this, and we've also, through recent data, have found that this is likely due to oxidative stress that is caused on the bacteria.
Thanks so much, Katie. That is that's so interesting. Dean Millen, do you have any questions?
You know I do. I always do. Katie, it really is fascinating. I'd like to start with the compound. Is it Perogalo? Am I saying that correctly?
Yeah, Perogalo. Yep.
Is that something that emerged from the literature that you started there, or how did you know where to start with compounds to do your investigations?
Yeah, so there is some evidence in literature about Perogalo, but it had not really been tested in biofilm formation. And because that is such a big factor that bacteria have to cause infections, we thought we would try it, and we got really positive results. So yeah, it's very exciting.
It is exciting. And so as you're looking at the hospital environment, I'm wondering, does this work translate beyond that? I'm thinking maybe locker rooms in athletics, or, there are other environments in which this would have, immediate value.
Mm-hmm. Yeah, definitely. So this could be used as a potential disinfectant, or depending on how toxic it is to human cells, as an antibiotic. The question is just, in terms of if you are applying this as a disinfectant in locker rooms, you could also be potentially eventually breeding further resistance. So I think it's important to understand in which situations, like in a hospital, obviously it's really important everything is clean, but you kind of have to pick and choose where you will use this.
And then, Amy, I always have one more. Can I keep going?
Go for it.
So beyond this compound, what's the larger lesson learned about fighting, the antibacterial? You got to forgive my science jargon here, but where do we go from this one compound to the larger lessons learned? Is really my question.
I think it's really important for everyone to realize that when penicillin was discovered, there was a huge boom in terms of survival from like everyday infections, and I think we're starting to get to the end of the antibiotic era, where it has been so prescribed for times when it's not necessarily needed. It's in our food and it's in our water supply. So it's something that needs to be addressed if we want to avoid starting to die from regular infections that we have not died from in about 100 years.
When my kids were young, which is a long time ago, whenever they had the slightest hint of an ear infection, they were given the amoxicillin or some other version. At some point, people got nervous. Are we giving them too many of these antibiotics so they won't be able to fight them when they need to? It sounds like that's related to your research.
And Katie, we do have a question from our live Facebook chat as well. And that's, is there any more information regarding the oxidative stress and what pathway may be disrupted? Sounds like one of your faculty is asking if you remember what they taught you in their class.
I think they're showing off is what they're doing.
We have not looked at the pathway yet, but obviously we've done assays where we're introducing a bacteria to hydrogen peroxide, which is what is kind of produced through oxidative stress, and but we haven't really done further research into the exact like mechanism for how it works. We're just kind of like moving forward with that right now.
Great. Thanks so much, Katie. I learned a lot of things, but I also learned how to pronounce that compound, which I've been mispronouncing for many, many years as an organic chemist. I can't believe I did.
Well, I can tell you that's the first time I've ever uttered the word in my life. So
I'll quiz you later, Dean Millen. So we've seen several presentations from our student scientists, but of course we're the College of Arts and Humanities, and we have some really engaging and vibrant work going on in our humanities and arts fields. And so we're going to switch gears a little bit and hear from Maggie Miller, who is an art and design media graduating senior, and she has been working with Stephen Burt. This is part of her capstone art project, and normally we'd have a nice gallery exhibit in one of the galleries down in downtown Biddeford. We have to do things a little differently this year, but I'm sure her presentation will be no less engaging. Her title is Trailblazers and Game Changers: Black Women Who Paved the Way for Their Modern Counterparts.
Thank you. Let me just pull it up. So my name is Maggie. This is my presentation. So this is revised work from last semester, as she said. It's the same topic, same motive, which is to give Black American women the recognition that they deserve. So this is my series. It was in the campus center most of this semester. And yeah, I pretty much just revised it, made it a little better, made it more me. So here is my artist statement, which I'll read to you. So the George Floyd protests of the summer and the larger Black Lives Matter movement has caused me to reassess what the purpose of art is to me and what role it has to play in my life. Many women of the past worked to pave the way for us to be able to do what we do now, whether it be poetry, politics, or sports. Women never had the place until one spoke out and did it anyway. We were only beginning to speak and hear about these women, especially Black American women who made history. My goal in this series is to not only educate myself, but the communities I am in, my hometown and my college. Not only are we standing together as we must, but we can educate ourselves so that we can understand the importance of this moment in time. We cannot imagine the summer of 2020 BLM protests are over, and we cannot stop talking about it. For me, I can use my voice and educate with art, which is why I was so inspired to create this series. I am able to celebrate the accomplishments of these incredible Black American women and express myself creatively, experimenting as I go and learn. So I am just gonna go through some of my slides, just give you some examples of what I've been working on. I'm not going to show all of them because I'll save that for later. But so this is Phyllis Wheatley. She's a good example of a trailblazer. She is the first Black American woman to write a book of poems. And then comparing this to Amanda Gorman, who you may recognize from the presidential inauguration. She is the youngest woman to speak at an inauguration and most accomplished young Black woman poet in America. And she wouldn't have been able to become a famous poet without Phyllis Wheatley and other people paving the way. So another example, this is Anne Lowe. She is the first notable Black American woman fashion designer. Then comparing that to Carly Cushnie, which you may recognize. Cushnie is a pretty big name. She is one of the most accomplished Black women fashion designers in this era. She wouldn't have been able to become a famous fashion designer if it wasn't for like Anne Lowe and that generation. So if you want to see the rest of my paintings, and I can go more into depth, come to the presentation. But yeah, thank you.
Maggie, that is wonderful. Amy, I'm just diving right in. You just stop me when you have to.
Go for it.
Maggie, when I look at your work, there clearly is a commonality across the works that you showed us, but I also saw, I think, some variation maybe in some of the media that were used. It looked like some had some actual three-dimensional work to them. Can you talk a little bit about how you actually created your pieces?
Yeah, so I did it all on unstretched canvas. I actually did it right behind me. So I wanted to just kind of experiment. So my Ann Lowe piece, I sewed the hat on because I think that's a good connection to fashion designers. I was just kind of having fun with it, experimenting. And actually my entire series that's up in the Commons, it's all sewn. So like it's a big banner. So I just wanted to make it more powerful in that way. And I also used acrylic paint. And then I also, on top of that, used watercolor markers and oil pastels to give it some more texture and some more layers to it.
Yeah, and that really came through on the screen, so I can only imagine how powerful that must be in person. I hope to make it myself over there to take a look. Tell me about the women that you chose to focus on, and how did you choose them? And did you work with Dr. Burt in that, or is that from your own research?
I kind of just thought about, like, I honestly just like went on social media. I just like looked up random people on Google, like, and then things would just pop up. Like I had no intention to do fashion designers or make it more like an art related thing, but I just really found this interesting connection, which I'll show in my presentation, about just like gymnasts and poets and ballerinas that I did. I just love the movements and I like painting that. So that's kind of what pivoted me to do more of that work. But at the beginning, I was just kind of like, I saw the inauguration speech that Amanda Gorman gave. I was super impressed, as everyone else was, and I really wanted to research that. And then I just kind of fell into a hole of researching all these incredible Black American women.
So out of all these incredible Black American women, was there any one or two in particular that inspired you or that you hope to learn even more about?
I honestly really enjoyed learning about the ballerinas because it's such a white dominated like career path and sport. So it's just like learning about that was so interesting, and like the women were honestly pretty hard to like find and research because it's mostly just young people now, because they didn't even allow people into like the American Ballet Theater that were like a Black American or anyone of color really until like 2015. So it's like insane. So I really want to learn more about that stuff.
Yeah, I don't know if you looked into music at all, but there were some orchestras that started doing blind auditions where the musician would actually perform behind a screen because they found so much institutional bias in the selection process for musicians. Certainly that wouldn't work in dance, but it does speak to the larger pattern that you're talking about.
Oh yeah, definitely.
Well, that's wonderful. And it really does show the scope of the kind of work that our students are doing, from the traditional lab research to the creative arts and everything in between. So thank you so much for sharing that with us.
Of course, thank you.
Thanks, Maggie. That's so interesting, and I noticed right away that you know, most of your the women that you chose were in some sort of art or creative field, and I too wondered if that came from your own influences. I mean, for me, I would have automatically, as a chemist, would have gone to think about scientists, and that's sort of my realm. So I really appreciate learning about these inspiring Black women. And Dean Millen pointed out that we do see such a wide array of work that we do in the College of Arts and Sciences, you know, from creative arts and you know, lab work and field work, but we also have a rich scholarship in the humanities, whether that's archival work going on by our historians. We've got faculty in philosophy and who are doing creative work in literature and all these different fields. And we can't showcase everybody here, but there's certainly lots going on. But we are going to dive, pun intended here, dive into some work from one of our marine biology students, and that's Ben Lafreniere, who has been working with John Mohan, who's one of our new faculty, and he's going to tell us how vertebral chemistry distinguishes nursery habitats of juvenile shortfin mako sharks. Take it away, Ben.
Thank you, dear Kierstead. And Let's do it. All right, so thank you everybody for coming. My name is Ben Lafreniere. I work with Dr. John Moham. And like Dean Kiersten said, we looked at juvenile shortfin mako sharks. Juvenile shortfin makos, they're very vital to the ocean systems. Mako sharks are ecologically important apex predators, specifically in Southern California where this study was conducted. And the problem is that there's not a lot that's known about juvenile shortfin makos. So this is why we chose them for this study. So the makos for this study, they were sampled opportunistically at two different sampling regions, one in Southern California and one in Baja California. Juvenile makos utilize these coastlines, both for their protection from the open ocean and the availability of prey species, which is why they're able to be found here. So the vertebrae that were sampled, like the one shown here, basically we can use these vertebrae to analyze the life history of the animal. Vertebrae grow with the animal, similar to how growth rings grow on like a tree. So that's why they're very useful for studies like these. So vertebrae would we've cut by like a transect line, like the one shown here, and it will give us something that looks like this. So all vertebrae have a distinct edge band, which you can see right there. And this band contains elements from the recent life history of the animal. And this can give us a lot of information about them. But some of the individuals we sampled as well were a little bit larger, so they had a distinct birth band as well. So this gave them us information about them shortly after birth that we could then compare to the recent life history. So the first statistical analysis we conducted compared elemental concentrations in the edge of the vertebrae versus the two sampling regions, Southern California and Baja California. And this basically helps us determine if there's any regional variation. We found that zinc, strontium, and barium were statistically different between these two sampling regions. And this tells us that basically these two habitats are distinct and it's showing up in the vertebrate chemistry. This can be things like upwelling and different ocean chemistries that are driving these elements to occur differently between the two sampling regions. Next, we analyzed the birth band elemental concentrations and the edge vertebrate concentrations to see if there's any shifts that are occurring as these makos grow. Is there things that are changing within their life that will make their chemistry appear differently in the vertebrae? And we found that lithium, magnesium, and zinc were statistically different between the birth band edge, which we showed here. And basically what that means is that something is changing within these animals. It can be things like diet, things like choice of habitat that are driving these elements to change. So the application of this research and this type of research is still very new. So there's really that's a lot known. It's never been done on juvenile shortfin makos before. So we know that these elements are changing, but actually what does that mean is still something that we're looking into now. But this data can help fishery managers and policymakers better protect these animals that are very, very threatened in their ecosystems right now. And we hope that future studies, both in the field and potentially in lab controlled studies, can help understand how these elements are actually changing. Thank you.
How cool is it that we get to talk about sharks after talking about fashion leaders in the industry? What a great morning this is. Thanks, Benjamin. You might have answered my question with one of the last things you said, but I want to ask it anyway for clarity. See, you did find chemical differences between the two locations.
Yes.
And you found some statistical differences. But what's not clear is the so what part. Is one healthier than the other? Is one more vibrant? Does one suggest a concern where the other one doesn't? Or for the moment, is it just differences that we're looking at the nature of those differences?
Yeah, so basically what we can see is that certain elements have been show variation based on different variables. So things like strontium has had a lot of temperature implications with it. So we can see, which would be expected between those two regions, that temperature variation would occur. Things like barium has been tied to upwelling, which is an ocean system. So basically we can draw that one ecosystem is experiencing more upwelling than others. It's not necessarily good or bad. We did look at some like heavy metals like lead and things like that, which we didn't see any difference. So there's no cause for concern or anything like that. But basically we see those differences and we want to know more why.
Right. And do you know if the two areas have any migration between them, or are these distinct and separate populations?
Yeah, so adult makos, they move very vastly over the ecosystem, so it's very tough to understand their specific habitats. But from the research that we've seen in past literature, we've seen that those nurseries have been fairly distinct. And we think that these findings can actually prove that they're actually staying in that system. And then potentially with those individuals that have a birth band and an edge, that we can see that maybe some cross migration is actually occurring.
Very cool. Now, a very, very different kind of question. How did you guys get the samples? I mean, what's the process by which you actually get this stuff?
Yeah, so mako sharks are a very vulnerable species, so we didn't want to simply go out to try and catch them. These were all opportunistic catch, things like bycatch from fisheries. We had some people that were into recreational fisheries that maybe they catch a mako shark and it doesn't survive on the line. So we did have not a very large sample size for this study. We had about 20 makos in it, but it was basically all opportunistic catch, and we wanted to basically make use of these animals.
And I just have one, it's really more of an observation. I'd love to hear your thoughts on it. You know, the public has a fear of sharks going back to Jaws, was that 1973, whenever it was, and we had this whole, and it seems to me that the more the general public becomes afraid of sharks, and last year we had a very unfortunate situation not far from here, that scientists become even more interested. It's almost like this inverse relationship between the amount of fear in the public and the amount of interest among this science community. Is there something to that, or am I just making that up?
Oh, I think it's very fair. Yeah, I think especially up here in the Gulf of Maine, we've seen a large shift of great whites that have been coming up here. And as much as people have been terrified about them, they still actually want to see them, it just shows that we have a very healthy ecosystem up here. And definitely in my experience, I always see whenever something like that happens that I start to get a lot of questions about those things. So it's very exciting stuff.
Yeah, very cool.
Thank you so much, Ben. And, we heard from Emily how our unique location has allowed her to do her research. I've never worked at another university where we've gotten an email from our director of safety and security saying, there's been a shark in the water, look out for that. Snowstorms and sharks. So Dean Millen, we've seen so many interesting research projects already, and most of these have been from students who are working with faculty on a research project or scholarship project. But we also have research experiences integrated into our courses that students take as part of their regular curriculum. And that could be, ranging from our research methods courses that we see in some of our psychology and animal behavior courses, or even course-based undergraduate research experiences like we've seen in some of our chemistry courses, where students are working on, learning the techniques through research, or maybe it's helping one of our history faculty with archival work and then putting on a museum display, or out at Freddie Beach, doing a dig with our archaeology professor. So certainly lots of interesting things going on. And that's a great segue to our next project from Paul Guglietta, who is going to be presenting his work that he's done as part of Dr. Gordon's research methods course in sport and recreation management. And his title is Student Athletes and Time Management. How can we help athletes help athletes?
So hello, everyone. My name is Paul Guglietta. I'm a senior sport management major here with a double minor in business and coaching. My presentation here is titled Student Athletes and Academic Success. It's a little different from time management, but obviously time management is still going to be incorporated into my study. My overall research question is, how can academic services at respective Division I universities unearth the real struggles that student athletes face? So I just wanted to focus on Division I athletes, but obviously uni is Division III school, which can also, a lot of these common themes and concepts can be pulled from Division I schools and integrated into the Division III nature as well. So Division I athletes are exposed to a wide variety of academic services, including academic counselors, peer groups, and even meetings with their coaches. They oftentimes tend to struggle during their freshman year of school, but after freshman year, several student athletes are still struggling within the classroom. And so despite all of the available resources, they still finding ways they're not keeping up academically sound. So I discovered two common themes in my research throughout my research methods class. First was that student athletes at such a high level are just simply overwhelmed with the busyness of their schedule. And this also applies to the Division III as well. Practices, lifts, competitions, all retaining through their sport, take up countless hours every week. And then oftentimes academics can find the back seat. Finding ways to manage time is often a difficult thing to do, but finding a solution to this will make student athletes much more successful in the classroom. It's just been proven. And secondly, what I focused my research on primarily was student athletes and academic services don't have strong enough relationships with each other to the point that one another are comfortable sharing everything going on in their lives. So it tends to be a lot of, shadowing. There's a lot of things that student athletes tend to hide from academic counselors and their coaches and stuff like that, that they're struggling, which is holding them back in the classroom. And that's what I did my research on. I wanted to unearth what challenges they were facing and ways that we could create those relationships to be better with each other so that student athletes can have an overall better academic experience as well.
Oh, that's great. Thank you so much. You're an athlete yourself, is that correct?
Correct, yes. I played football here for 4 years.
And from my position as dean of the college, it's really important that we use the term student athlete and not athlete.
Absolutely. Education's always first.
And when you look at Division I, we know by looking at whether it's football or any of the major sports within NCAA Division I, we tend to see that inversion, right? Even though we call them student athletes, the reality of the situation is they're, for some of them anyway, they're athletes that are studying and not students that are being athletic. Yes. Does your work speak to that and how we might find a better balance of sustainability for these students so that their education really does come first?
Absolutely. I think that's one of the most important things I wanted to touch on was creating a better academic experience for student athletes. I know that, at the Division I level, oftentimes their majors get picked and changed for them. And I think that that's wrong. I think that they should be able to pursue the education they want. They should, their number one goal should be to be a graduate, not just be a student athlete. It should be academics first.
And I'm going to ask you a question that may surprise you, but since we're on the topic, what are your thoughts on student athletes in NCAA Division I schools being paid, or at least now having rights to monetize their own name and likeness?
Absolutely. I understand both sides of it. I stand on the side where I don't think they do. I think that having a lot of them at least have full scholarships, they're having their education paid for. Although I think that there should be some more regulations in terms of, like I previously addressed, on them not being able to choose their majors and, getting a general studies major. But I think they should still have their full ride scholarship, but they should be able to choose the major and the degree that they want. Because after all, less than 1% of college athletes are going to be going professional. And, even after that, your career is over by the time you're, 30, 35 years old, and they need a life after that. They need to have an area of concentration that they're interested in, that they want to, have a life in just outside of their sport.
Yeah, I could not agree with you more. And I think that's such an important message, not just to the college students, but the high school recruits, so they understand going in the importance of that balance that we've been talking about. And I'd like to think that at UNE, we've achieved that semblance of balance, perhaps better than some other institutions, and you would know better than I would. So I'm wondering, you looked at D1, but having lived through D3, how might your results have differed?
I think at Division III, I think there's a lot of practices and lifts and stuff like that, but a lot of these high-end Division I programs, like I did my proposed research for the University of Georgia, for example, their entire lives are just circulating the athletic facilities, and their dorms are in the athletic facilities. They do their classes and their work in the athletic facilities. Like they're just always monitored by athletic administrators and coaches. So they live and breathe their athletics, where at like a Division III school like us, we have flexibility to do things outside of our sport, and we're able to go to different parts of campus, and we have social life with every other student, and it's different than just having your whole life revolve around that sport. Our lives definitely makes it easier to have our lives revolve around our education instead of just our sport.
Yeah. And Amy, can I sneak in one more quick question?
Of course, you're the boss.
Paul, who's your team for the NFL?
I'm an Eagles fan.
So what'd you think of the draft last night? You guys made some strange moves.
We did, but I'm happy. I'm really happy that we drafted Devontae Smith. I was very happy about that.
Very good. You may not know this, but I just moved to the region after living in Philly for about 30 years, and but right not on. An Eagles fan. Not an Eagles fan. But that's okay. We're still good. We're still good. Thank you, Paul. That was wonderful. Really, really insightful.
Thank you.
So before we move to the next presenter, who is Mariah Bershelsky, one of our neuroscience students, Dean Millen, you you speak with a lot of prospective students and families and represent the college with our board of trustees, for example. And what would you tell someone who was asking in general about, if they asked about the benefits of undergraduate research?
Yeah, that's a great question. Thanks. And I'm going to start with just a very quick story. When I arrived in the summer, I think I started on July 1st. And one of my first goals was to meet the students in CAS at UNE, because I hadn't yet met students. And the summer undergraduate research experience was going on at the time, the SHORE program. And I was able to have lunch with, I think it was 4 students who were immersed in their summer research. And most schools make research available. So it would be disingenuous to say, if you don't come to UNE and study in CAS, you're not going to do undergraduate research. The difference at UNE and the value of our education is the real hands-on, independent nature of that work. Our students aren't just doing the work. They are learning to present their work, to talk about their work. They understand their work in such a way that they're conversant about it. And I've heard time and time again, and I've seen it firsthand, that our undergraduates speak and perform like graduate students. The way in which they deeply understand and appreciate the work that they're doing, it's not like they're looking from the other side of the lens. Their sleeves are rolled up. They're getting their hands dirty, regardless of if it's dirty with paint or dirty with kelp or dirty with data. Our students are just immersed in a way that you don't typically find. And as you said, it's not just in these independent projects, but it's embedded in the coursework. So it surrounds us. It's, if you let me use the phrase, it's in the DNA. And it's really difficult for students to graduate without having had some kind of firsthand experience. And going back to Nicole, and students like Nicole, when they start early, it's woven into the fabric of their entire 4-year experience. So it's qualitatively different at UNE and CAS.
Yeah, I would agree with that. And I can say I transitioned into, my career in my graduate program, having done undergraduate research. And so it's wonderful that our students have these opportunities. But with that, we'll switch over, and our next presenter is Mariah Bershelsky. Mariah is a graduating neuroscience student and the recipient of this year's Undergraduate Student Research Award in the Natural Sciences. Congratulations, Mariah. Mariah works with Dr. Berman as well as being part of the Berman Collaborative and the Center for Excellence in Neurosciences. And Mariah is going to tell us about her really interesting work. Amygdala corticotropin releasing factor signaling is required for later life behavioral dysfunction following neonatal pain. Take it away, Mariah.
Thank you, Dr. Kierstead. I would love to tell you more about it. So this data is based upon the most recent publication to come out of the Berman Collaborative this past March, following discovery that neonatal pain followed by juvenile trauma produces a robust hypersensitivity to tactile pain. We choose to investigate this since newborns who spend time in the neonatal intensive care unit, or the NICU, are more likely to develop anxiety, depression, and have altered pain thresholds later in life, making this a very important field of study. Previously, we found that neonates experiencing early life pain demonstrate an immediate change in expression of a neurotransmitter called corticotropin releasing factor, or CRF, within an important brain structure called the amygdala. Few other brain structures or neurotransmitters are affected at this point because they are still developing. In this project, we seek to examine whether lasting changes in CRF expression within the central nucleus of the amygdala is a potential mechanism of the effects of neonatal pain on later behavior. And I apologize if I'm cutting out. I just got a notification that said that my internet is unstable. So as we continue, to examine this, we use a neonatal manipulation protocol on rats that mimics the heel lancing that occurs in the NICU to gather blood samples from premature babies. When looking at these graphs, the x-axis has the neonatal conditions the rats undergo, and the y-axis has what we call the Paul withdrawal threshold. This threshold is measured in the amount of pressure applied in grams, meaning the higher the threshold, the more pressure from a von Frey filament or a fishing wire-like filament was applied to the rat's paw to elicit a pain-like response. If you look over at the first graph on the left, you can see that both a painful needle prick and a non-painful handling for the first 7 days of life creates hypersensitivity that we have previously seen, with these red and yellow bars being significantly lower than this blue bar. On the next graph, you will see that CRF receptor 1 antagonist antialarmin is shown to reverse the hypersensitivity, with these darker bars being significantly higher than these lighter bars of the same color. Then as we continue on to the rightmost graph, you will see that CRF receptor 2 antagonist atriosin 2B did the same with an even more interesting effect, leading to an overall increase in sensory threshold, which corresponds to lower pain for all groups, seen through the dark red bar being significantly higher than the dark green when collapsed across the neonatal condition. From these data and more, we have concluded that the CRF system may be a viable target for treatment looking at reducing the subsequent fear and anxiety produced by early life pain.
Wow, that's great. Thank you so much. One of the things I remember learning myself is that pain is a good thing, right? And so if I reach toward a hot stove and my finger gets hot, pain helps me to pull my hand away. With studying neonatal pain, is there a threshold at which pain is healthy and a good thing? Where is that line between the benefits of pain and then the as you said, can induce stress and anxiety and all these other negative attributes?
Yeah, that's something that we haven't quite looked at. As you said, like pain is a good thing, at least later in life, because we're able to respond to painful stimuli and protect ourselves. But as babies, we're modeling the neonatal intensive care unit. So they're undergoing all of these painful, necessary processes to keep them alive and well. But they typically undergo them without any kind of pain relief or anesthetics because of the fear of the long-term effects of that. So it's an interesting concept of trying to find like what might help build resilience, as a lot of people call it, and what might be too much to create that adverse effect later in life. And that's definitely something that could be looked at. Maybe if you modify our neonatal protocol, instead of doing 4 needle pricks a day for the first 7 days of life, with maybe reducing it to 1. We've also found differences between maternal separation, so the rats being separated from their mother for the day instead of going back to the mother. All very interesting research.
So given that there is so much that you could possibly study, how did you hone in on this one particular question?
So that would have to be to Dr. Berman primarily. All of his research is based around this idea of neonatal pain. And so we're currently in like the second phase of our research, and that's we've previously shown that there's a hypersensitivity. So now we're trying to find out what the biological and physiological reasons for this is. So we look at the specific receptors in the amygdala, because the amygdala is known to be about fear and pain processing in the body. And then we look at these specific receptors for CRF, because we know that the CRF system plays a role with pain and the adrenocorticoid axis and the positive feedback loop with that. So it was a kind of an easy choice there. There are a couple other targets that other labs that we work with look at as well that are also viable targets. But we chose to hone in on this because it was our most promising outlook when we started.
And Amy, Mariah's answer is indicative of what I said a moment ago, but you can tell the immersion and her ability to just talk about her area of study off script, if you will. It's just so impressive. But one last question I have. For the layperson, you're studying rats and you're talking about neonatal humans. Convince me that what you've learned about a rat actually translates to a human being.
Yeah, so we use rats as a model organism because their brains are structurally comparable to humans. So as a useful model organism, they're the mammal, and they have pretty much the same structures in the same layout of the brain. And they have the same kind of hormone processes that we do, maybe on a smaller scale because of how small they are. But we can attribute them to each other because we can find out these processes and then look at humans later in life. But this allows us to do all of the things to the rats so that we're not hurting babies, because that would be very unfortunate.
Thank you so much.
Thank you, Mariah. That's so interesting. And I thought of your work almost just a year ago when I had a baby, and I was in the hospital and they were doing tests on him, and he wasn't in the NICU, so they weren't very many. But every time they wanted to do something, I thought, what about that trauma? So your work has had an impact on me as well. And Dean Millen, I've got our Facebook page open, the UNE Facebook page, and I'm watching the chat, and the chatter is about how talented our students are. But we're seeing some comments from our faculty about, they've had a student in one class and they didn't know they were involved in this research, and they're so talented across a broad array of areas. And I think that speaks to what we hope for our students at UNE and the College of Arts and Sciences. We don't want our students to leave. We want them to be experts in their discipline for sure, but we want them to be well-rounded and have this broad knowledge and contextual knowledge as well about where they fit in the world and how they'll contribute going forward. And that's part of our core curriculum. That's part of our general mindset with our experiential education as well. And so this research symposium webinar is one slice of what I think we offer in the College of Arts and Sciences.
Amy, how many students are actually presenting in different formats and modalities today across the board?
We've got about 100. So we've had posters up across campus all week for people to view. And then starting 2 minutes ago, we're having some live Zoom sessions. We're running over because we just can't help ourselves. And but we've got over 60 projects representing every school in the College of Arts and Sciences. So, absolutely. So without further ado, because I know we wanted to jump to the next parts of our day, and I'm super excited to hear from Virginia May, who is a freshman studying environmental science. She's working with Dr. Tom Clack, and her project, she's part of the chestnut team. And so her title is American Chestnut Restoration, Transgenic Blight-Tolerant Pollen Production and Analysis. Virginia, the floor is yours.
Hi, thank you so much. Yeah, let me just share my screen. Okay, as you can see, this is actually a photo of our success. That's our blight-tolerant pollen on the catkins of one of our American chestnuts. And I'll give you some background. So American chestnuts was once the dominant tree of the eastern United States forests. This redwood of the East was a keystone species that could mature up to 12 feet in diameter and over 100 feet tall. So every 1 in 4 trees of the American eastern forest was an American chestnut. That's how like spread out it was. And its range was from Maine to Georgia. And so, however, about 4 billion trees were lost to an accidentally imported fungal blight. So that's what the American Chestnut Project is all about, is about restoring this amazing species. So in UNE's efforts to bring back this iconic species, our team has successfully sped-bred blight-tolerant pollen. So speed breeding is a very intensive growing process, which allows us to produce pollen about a year after sowing the seed. So in the wild, a sexually mature chestnut would take about 5 to 6 years to get pollen out of it, but we're doing it in just around a year. And so because of this very intensive growing process, only some of the plants are able to produce pollen while the majority of them are not. And so this this production of pollen per seedling is highly variable because of their genetics. And so 51% of all of our pollen came from just 2 of the 24 seedlings. 11 produced any pollen at all, and 13 were not actually able to produce pollen. And so last summer, in the summer of 2020, because of our efforts, over 5,000 fertile chestnuts were produced over 7 locations across the country with our UNE pollen, which is it's a huge step because with help across the nation, UNE is leading the transgenic pollen production that supports the entire American Chestnut Project. And it's a huge step forward to returning this like quintessential American species to the rest of the United States and back to its natural habitat.
So thank you, Virginia. You just called it the quintessential American species. And so there's a nostalgic feel to this, like we're bringing something back that we had lost. But beyond the nostalgia, what did we lose by losing the chestnut? What, is there something unique about that tree that will bring something back to our ecosystem?
This was actually the main lumber tree of the eastern forest. So because it was a canopy tree and could grow up to 100 feet tall, it had a very specific ecosystem role, but also is a hardwood that was made, it was called a cradle to grave tree. So this was lumber and a major food source for also the people of like Appalachia and surrounding areas. So it was really a part of everyday life before it was almost functionally extinct.
And was it a strong tree in terms of, well, it almost went extinct, right? And so was there something about its own makeup that made it more susceptible? And part B to that question, are you confident that this time around it will be able to sustain the many pathogens and challenges that yeah.
So the only reason it was susceptible to this blight is because this was a blight that had functionally like evolved with the Asian species of the chestnut trees. So kind of like a cousin, different species. And so it was able to latch on easier to the chestnut trees and kill them faster. And it had no tolerance to it. And so with this tree, it has all the same genetics as a regular American chestnut, but it has one inserted gene, and that's a wheat gene. And it produces the oxalic acid that's able to fight against the blight. And so it actually has shown that it's how the genetics are passed down, and we're able to fight against the blight. It makes it tolerant, doesn't make it 100% resistant, but it gives it a much better shot to be able to survive back in the wild. And actually it shows that it has the same functionings as a regular American chestnut tree in the wild. So it's actually very healthy for the environment. A lot of testing was done on that.
And you said in your presentation, it was a quarter of all the trees. I forget the exact number that you told us. It was a quarter.
About 1 in 4 trees was an American chestnut in the eastern forests like 120 years ago.
And so if it thrives and if it starts to repopulate our forests and the like, what trees will we lose as a result of its reemergence? Is there a concern of maintaining balance of other tree species?
No, actually, because when the chestnut disappeared, it was red oak and maple that actually became the predominant species that was able to take over. And they produce a lot of ecosystem services, but nothing like the niche that the American chestnut can really fill, because it was even during the testing to make sure the transgenic trees were safe enough. It was just even as small things as like the leaf litter that it produced made the tadpoles grow faster, because they evolved with the American chestnut it's a very nutritious and fast growing tree. And it's like the environment's missing this keystone species.
And one last thing, Virginia, I'm presuming that Dean Kierstead misspoke a moment ago when she identified you as a first-year student. I presume you're graduating in a couple of weeks.
I am a first-year student.
And again, there's a testament to what the uni experience has to offer. That's just wonderful. Thank you so much, Virginia.
Thank you, Virginia. That's so interesting. And I get a constant reminder of this project when I walk across campus and I see the seedlings that were planted with the special markers, or I walk by our greenhouse and see it up at night, and there's that work in action. So thank you, Professor Clack and all of his Team Chestnut researchers. And thank you to our student presenters that were here today. I've learned so much just from these short snippets and your engagement with this. Thank you to our audience who joined in. Thank you, Dean Millen, for asking such insightful questions. And of course, thank you to our communications team who helped us put this on.
And if I can just add, and to the faculty mentors who worked with these students and continue to work with these students, your work is deeply, deeply appreciated.
Absolutely. Thanks, and have a great day.
Bye, everybody. Thanks, Amy.